Hello again. This is just a continuous of the previous one of the protein transport. I just want to make it a big video. So there are two pathways in the cell when you're transporting protein within the endomembrane system. There are two things you can do. One of them is the exocytosis, which means you can take the protein from inside the cell and take it to the outside of the cell. Or you can do the reverse. You can take something from outside the cell and take it inside. So both of them are working. They have different purposes. It doesn't mean that one of them happens only in one cell. We are going to see them one by one. The first one we are going to see is the secretory pathway, which means it's going to be exocytosis. Remember, ER is the place of production, so all the protein production take place here except the cytosolic proteins. But of course, the cytosolic proteins, they don't ever enter into the endomembrane system. If a protein is translated here in the cytosol, it can end up maybe in nucleus. Say it can go to nucleus through the nuclear pore complex. It can maybe transport it to the mitochondria. It can maybe enter into the peroxisome, but it's not going to be part of the endomembrane system. For a protein to be part of the endomembrane system, it has the ER start transfer signal, whatever, no matter what, and then it can have the stop transfer and start transfer again if it's going to be a transmembrane protein. If it's going to be a lumen protein, it doesn't have to have any kind of stop protein or not. But if a protein is going to enter into the endomembrane system, it should contain an ER start transfer signal. So that's the ticket to the endomembrane system. So once the protein is translated into the ER lumen, let's say it's here, or let's show it with a different color. It's not going to be a random movement. It's not going to diffuse into the other organelles. This is actually quite well regulated, quite well controlled. Which protein is going to be taken to the next organelle and how and when is well regulated. Now, first, when a protein is translated here, the conformation is going to be checked. The conformation, of course, requires the shape. It means the shape. So the primary structure is coded by the genetic information. The primary structure means the amino acid sequence only. It comes totally through the genetic information. There is the secondary structure, which is the alpha helix and beta sheet, which is again usually determined by the genetic information. And there is the tertiary and quaternary structure, and finally the conformation is taken. This quaternary structure maybe requires different polypeptides to come together. It requires maybe the post-translational modifications, maybe some methyl groups will be added. So some post-translational modifications will be applied. The post-translational modifications, they start here and they continue here. So once it's leaving the Golgi, the protein is mature and ready. But all through way, whenever it is translated from ER to cis Golgi, by the way, this part is called cis Golgi. And when it is transferred in between and when it is transferred to trans Golgi, at every case, it is classified, it is controlled, and it is modified. The first thing is the disulfide bridges, which means sulfur to sulfur bonds. I said that in the biological world, we only have the covalent and non-covalent bonds. The covalent bonds are strong, the non-covalent bonds are weak. So this is a covalent bond and a strong one. It happens between two cysteine amino acids when because they have this free sulfur. This sulfur to sulfur bridges, these covalent bonds only happen in the ER conditions because only the ER has the oxidizing conditions. It's only oxidized here in the ER. If the same protein is translated here in the cytosol, the disulfide bridges will not be formed. Okay, so it's specific to here. And these disulfide bridges, they're important for taking the conformation. Without these bridges, the conformation is not fixed, it is not secured. Okay. And then the post-translational modifications start. One of the many common modification is adding the sugar groups, and these are actually carbohydrates. And maybe you remember the name. This is called glycosylation. Glycose is sugar, so it's just being added to sugar. And these sugars, they have many functions. These sugar groups, of course, when they reach to the plasma membrane, when they are here, they are going to give the identity of the cell, because we know that identity of the cell is mostly provided by the glycoproteins, because this glycosylation is cell specific. They can also show that where in the endomembrane system you are localized at, because the glycosylation here in the ER is different from the glycosylation pattern here in the Golgi. So each of them has its own glycosylation patterns. Even in the cis and trans sections of the Golgi, there are different glycosylation patterns. So glycosylation is, is a mark of localization. We can take it like a compass. It shows where you are, or you can take it like the Google Maps. Okay. It all starts in the ER. In the ER, even while it's already being translated, this is the, you remember this translocator? And here is the start ER signal. This is the growing chain. So as soon as it comes across the tripeptide signal, this tripeptide signal is a very small and specific signal containing an asparagine. From this dolicol, the entire 14 sugar group is transferred to the asparagine. And it is called an unblock transformation. So these glucose groups are not transferred one by one. All 14 of them, they are transferred all at once. So this is an unblocked transformation, and this is called N-linked glycosylation. So I'm sure you have seen more in biochemistry, so it's going to help you that way too. So this is what happens in the ER. In the ER, they are more or less the same, all the glycosylation, but it's not going to stay the same. Once it enters into the Golgi, things are going to start to change. Some of the sugar groups, they are going to be chopped off, and here some new sugar groups will be added. And you know that these sugar groups, they can have different bondings, different shapes. So they have more diversity than the, let's say, proteins. So yes, I can say that they are more diverse than proteins in the case of shape and bonding. So they are going to change step by step. Maybe in the next step, these two are gone, and maybe in the other step, some other sugars are added. So it is modified step by step in the Golgi until it reaches to the trans Golgi. And here, when it is leaving the trans Golgi to endosome or to plasma membrane, now it's the mature protein which is ready to work. I really hope that it's clear about the glycosylation. Glycosylation is the key for the function of many enzymes. Also, they are key to protect the proteins from the enzymatic activity, especially if you think about the lysosome. The lysosome is highly acidic. So if a protein is going to work inside the lysosome, it has to be highly glycosylated. Otherwise, it will not be able to be, it's not going to be protected from the acidic environment of the lysosome. What protects this protein from the environment of lysosome is the sugar groups. Also, I said that it gives the identity of the cell, but not only that, it gives physical protection. These glycosyl groups, they are interacting with lots of water. The water molecules, it makes the friction low, so it becomes kind of slimy. You know the slimes that children play. So especially if it's a cell like a white blood cell which is circulating through the body, it has to be protected from the friction force. And in that case, these sugar groups, carbohydrates, are very helpful. Okay, this is one thing about the ER. First, in the ER, modification or the post-translational modification starts. And second, in the ER, there's a quality control. Okay. In the ER, once they are translated, and now we are looking at the ER lumen proteins, these are not transmembrane proteins, the protein can fold properly like this, or it can misfold. Now, this misfolded protein has the correct amino acid sequence, but the shape is wrong. And how can you understand if the shape is wrong if you were in the molecular environment? Well, if you see some hydrophobic amino acids which are facing outside, it means it's the misfolded proteins, because the hydrophobic amino acid, they should be kept inside. It should be hidden from the water, because these hydrophobic regions, it can cause the proteins to accumulate. It can even become prion proteins, and it can kill the cell. So they are dangerous. So whenever there is a misfolded protein, the chaperone proteins help them to get the new shape. The chaperone proteins kind of push them to lose the shape and get the shape once again. So they're going to get the correct shape. And if they cannot get the correct shape for a long time, if it is tried and still not taking the correct shape, it's going to be pushed to the cytosol, and it's going to be degraded by the proteasome complex. Yes, it looks like a waste of material, but keeping the quality high is at utmost important. So if a protein is misfolded, even slightly misfolded, just get rid of it, because these misfolded proteins can be very dangerous in the late future. You know the cystic fibrosis. In that case, for example, there is the chloride channel, which is mutated, which is slightly misfolded. It can function still perfectly, but just because it is slightly misfolded, it stays in the ER. It never goes the way until the cell membrane, because there are many checkpoints. It's going to be checked here in the ER, it's going to be checked here in Golgi, and several times. It cannot pass through these checkpoints, so it can never reach here where the chloride channel is required. This is why these people are defective. Okay, what happens when there is a problem with the misfolding, when there are loss of misfolded protein? It creates a stress. We call it unfolded protein response. So when there are many misfolded proteins in the cell, when the proteins are not in the correct shape quite a lot, the ER enlarges. Because ER is responsible of this quality control, it has these chaperone proteins here. So a bigger ER means a bigger control. So this is the first case. So the ER gets large as a response to the misfolded proteins, and if it cannot control the situation, even with a very large ER organelle, the misfolded proteins cannot be corrected. Unfortunately, the cell suicides. It kills itself, because a cell with too many unfolded proteins can be dangerous for the body. So it has to kill itself, induce apoptosis, so that it's not going to affect badly the rest of the cell. And this is the case in diabetes. I'm going to explain it better tomorrow in the lecture, sorry. And this is the structure of the Golgi. This is the cis Golgi. This is actually where the ER is located. From ER to cis Golgi, the vesicles come and maturate, and then they move in both in tracts, and they also they're going with the vesicular transport, and then finally they find themselves in the trans-Golgi. The nice thing about the Golgi is that I'm going to draw it really badly. I'm sorry, mid and trans. Even if you totally delete the cis Golgi, even if there is no cis Golgi anymore, it's no problem, because Golgi is anyway a transient organelles. Soon, the organelles will come from ER. They are going to start to accumulate, and once there is a good number of vesicles, they are going to fuse, and they are going to form a new cis Golgi. And this is the natural course of things. The vesicles pinching off from ER, they form the cis Golgi, and they become mid Golgi, and then they become trans Golgi. And if you put a fluorescent marker, and if you watch the trans Golgi, what you call trans Golgi now will be gone in half an hour, or maybe let's say in few hours, because in few hours it's going to disappear. It's going to be broken into vesicles. Maybe this one is destined to the plasma membrane. Maybe this one is destined to the lysosome. This one is to the endosome. But anyway, it is not going to exist anymore. Instead, the mid Golgi is going to move and form the new trans Golgi. So let's draw step by step. This is what you see at time 0, at time 1. You're going to see some vesicles coming from the ER. The cis become mid become trans become new vesicles. At time 2, they will form the new cis. This become trans. And I'm sorry, this mid is now, this mid is now new trans. This is why I say that Golgi is a dynamic organelle. Okay, exocytosis can happen in two ways. It can be well controlled, like in this case, or it can be random. Every cell, time to time, exposes its contents to the outside. This is normal. This is also showing what is going on inside. This is called constitutive secretion. Regulated secretion has a purpose. Not everything has the regulated secretion, like when the cell is activated, when it's going to produce hormones, for example. It's going to do regulated secretion. And think about the neurotransmitters. The release of neurotransmitters to the, let's say in this case, it can be the synaptic cleft. Which one can be this? Do you think the neurotransmitters are released in a random constitutive way or in a regulated way? Of course, they're released to the synaptic cleft in a very well regulated way. They are regulated by the action potential. So this is a very nice example of regulated transport. And in this case, you see a hormone secretion, which is the insulin secretion, that is communicating with the body whenever they need this hormone. The opposite is the endocytic pathway. So in the small world, it can be one cell eating the other cell for food because they are feeding on each other. In this case, this is our white blood cell eating a microbe, a bacterium, because this white blood cell is trying to deactivate this microbe. Soon it's going to meet with the lysosome, and lysosome has lots of digestive enzymes, so this bacterium is dead now. Okay. Cholesterol is transferred to the cells in a similar endocytic way. Remember, cholesterol it's not a phospholipid, it's part of the, but although it's not a phospholipid, it's part of the membrane structure, and we need cholesterol. They have to be transferred to the cells, and because they are hydrophobic, they cannot be transferred themselves. Oh, something happened. Okay. They cannot be transferred themselves. Some other proteins called LDL, which is the low density lipoprotein, has to transport the the cholesterol to the cell, and they are not taking just by fusion, they are taking by endocytosis. So here the membrane is curving, and remember these proteins are called clatrins. The clatrins help the membrane curve. Finally they become a vesicle, and after that the clatrins shut off, just in the case of the Golgi example. Now it's the, oh god, now it's a free, okay. Now it's, oh, come on. I'm very sorry for this. Okay, I'm going to learn it. Okay, now it's a free vesicle. And then it's going to be an endosome, which means it's a membrane taken from outside. And it's going to meet with the lysosome, and once it meets with the lysosome, it's going to be degraded, the cholesterol gets free, and this free cholesterol is used up. You can read the story of LDL and HDL, why one of them is really good for you and one of them is dangerous when it's accumulated. Okay. This vesicular transport is also a way for the for some cells, a way of transportation, because remember these cells, like in the case of skin cells, like in the case of intestine cells, they have two surfaces, the apical surface and the basal surface. These apical and basal surfaces, they are different from each other. They have two different protein composition because there are tight junctions here, and these tight junctions, they're, how to say, they stop the protein movement. So the protein can move here and that way, and here too. It can move that way. It can even come here, but it can never pass through the tight junction. So it creates two different surfaces for these cells. Okay. Sometimes they can be taken from outside by endocytosis and can be used here, but sometimes a vesicle is taken from one surface and it's just released to the other surface. It's, it's a way of transport. Like if you're going to go to US, for example, but if you cannot find a direct flight, you can go to Germany from Istanbul to Berlin, and from Berlin to New York. In this case, the function of Berlin is nothing but a temporary transportation stop. In this case too, after the vesicle is endocytosed, it's just a temporary stop. It will directly go to the apical membrane, the basal membrane, because it is required in the basal site. I said that, okay, the story of endomembrane system. Let's sum up a little bit. It has been, oh my god, more than 20 minutes. The members of the endomembrane system contains Golgi, vesicles, lysosomes, endosomes. And out of this, the indispensable one is the ER. ER is the all start of production. Even if you get rid of all of them, ER somehow manages to recreate them. And here in the case of lysosome, for example, it actually is nothing but a vesicle. It starts its life as a vesicle, and then the protein pumps, the proton pumps comes here. This is the proton pump. And remember, pump is doing active transport. This is why it requires energy. The energy here is provided by the ATP. And using this ATP, the proton are pumped inside. Of course, when the proton comes inside, it's going to be acidic. And there are some acid hydrolases, like nucleases which digest the nucleic acid, proteases which digest the proteins, lipases which digest the lipids, etc. And this is a digestive organ. So whatever is taken inside, maybe there is an endosome which is just taken from outside. It may contain a bacterium, it may contain how to say, some random things from outside. These two are going to meet. Lysosome is going to meet with the endosome, and these proteins here, they're going to, once they meet, once they become fuse and one organelle, it's going to digest the bacterium here, like in this case. It fuses this organelle, this vesicle fuses with the lysosome, and the bacterium is dead. Okay. It's not always the extracellular stuff that we digest. Of course, we can take things from outside, like in the case of pinocytosis, and it becomes late endosome by meeting with the lysosome. But we can also do digest our own organelles, in the case especially about the mitochondria. Killing the mitochondria is very important, because if you do not kill the mitochondria, they will just get old. So you want your mitochondria fresh and healthy. If they don't become fresh, healthy, and fully functional, it's a way of aging. So old people usually have older mitochondria, because this autophagy of the killing of the mitochondria is slow in their body, while with the young people they recycle their mitochondria quite frequently. So the mitochondria is always young, always fresh, and always healthy. And this is the guy who discovered the mechanism, Yoshinori Oshumi. He got the Nobel Prize for that. You can go read the Nobel Prize